If there's no questions, let's go ahead and start. I've done some grading. I don't think I'm showing. Am I showing the screen? I don't think I am. I did, I think it was lab 4. Let me check on that. Why isn't that showing? There it is. Yeah, I did worksheet lab 4. And I did something last night and yesterday. Don't remember what it was. But I did the unknown notes. So I sent out the data for the unknown project last night. I did something else yesterday too, and I don't remember what it was. So if you have any questions, please ask. Oh, this is something I can plug. There's a number of students who have not started on the unknown project. You need to get started on that right away, or you will not have time to finish by the end of the term. So please get started on it. If you have questions about that project, email me or ask me during the lab if you don't mind other students hearing.
Okay.
All right, let's begin. Let me see. It looks like I've shut down the gradebook so I can show this. I don't think there's terribly anything I need to talk about, but oh, there was one point, and that is if you're late on getting, I think it's this week's worksheet for the lecture, all the questions have been answered. So just start. They're already starting it over. Go ahead and just do the next number. And yes, it'll be repeating, but I don't have anything else to do. So just do that. Today is October 4th, and we are supposed to be on, not October 4th, November 4th. We're supposed to be on chapter 8. And we are actually on chapter 8. So we're pretty much caught up. There is no lab today. I will log in for 15 minutes, but at 6:45, if there's no questions, I'm going to log off. Remember, your infectious disease project is due this Saturday. It must be turned in by 11:59 p.m. on Saturday, November 6th. I think there's an election going on at the moment, but I was trying to remember, that might have been needed to be turned in by last Tuesday. It might be this Saturday, I don't remember. Anyways, any questions about any of that? If not, I'm going to shut that down and begin on chapter 8. Should have got that open before class. All right, we had talked about, I think we got further than DNA replication, didn't we? Let me see if there's another two here. Yeah, I think we were talking about RNA transcription. Was that right? Did we talk about this slide? I'm not hearing any answers, but I think we talked about this slide. So let's talk a little bit further about RNA transcription. This is putting it all together in one step. The RNA transcription links, I've got a note saying it does not work. Let me check on that. Anyways, RNA transcription begins when the RNA polymerase binds to the promoter. The promoter is a region of DNA upstream of the gene. And that looks like this is working. So it's a region of DNA upstream. Nope, it's no longer available. Crud. The RNA polymerase begins there and then moves down the DNA. And as it moves down the DNA, it uses the DNA, well, it makes it single-stranded, first of all. And then it uses the one strand of DNA as the template for making RNA. And you can sort of see that here. Let me blow this up. Oh, that's way too big. Come on. There we go. So the RNA polymerase is moving down the DNA on this strand here and using that DNA, which is the dark purple, as a template for making the RNA. And the green line is the RNA. What the RNA polymerase does is it reads this template. And so it saw an A here. So it puts the complementary RNA nucleotide in, which I don't remember if we've discussed this or not, but the RNA complementary base to a DNA that's A is U. And then it sees the C here, so it puts in the G. And then it sees a T here, and it puts in the A. If you don't know, DNA, the four nucleotides are A, C, T, and G. And RNA has three that are the same. So G is the same, A is the same, and C is the same. But there is no T in RNA. You put in a U for uracil. I don't remember if we discussed that or not. Anyways, the RNA polymerase just reads down the DNA, transcribing RNA, and that will continue for the gene. But at the end of the gene, there's a region that is called the terminator, and it encodes for the RNA polymerase to fall off the DNA. So when the RNA polymerase reaches the terminator, it falls off the DNA. RNA transcription, of course, stops, and the RNA, not template, but the RNA strand then falls off the RNA polymerase. Any questions about any of that? Remember, only the genes are transcribed. Any DNA that does not encode for a gene product, meaning it's not a gene, will not be transcribed. We already talked about that. Once the RNA leaves the nucleus, if it's a eukaryotic cell, obviously it's in the cytoplasm of a prokaryotic cell. But once the RNA leaves the nucleus, if it's a messenger RNA, it will then go to the ribosomes and start protein translation. So the synthesis of proteins happens at the ribosomes. The messenger RNA is translated into amino acids to make the proteins. Just a minute. I was going to say I was going to sneeze, but I couldn't make it out in time. The tRNA and the rRNA, nothing further happens to those RNAs. They are the end product of the tRNA and the messenger RNA genes. And that was the point we talked about last time. How does the RNA polymerase know which RNA to make? And that is, it doesn't need to know because it's the gene that codes for what RNA it will be. If the gene codes for a protein, it'll be transcribed as messenger RNA. But if the gene codes for tRNA, it'll be transcribed as tRNA. And if the gene codes for rRNA, it'll be transcribed as rRNA. Any questions about any of that? No questions. Well, here's a question for you. What's different between the RNA of messenger RNA, tRNA, and rRNA? What's different between those three RNAs? Do they have the same nucleotides? Do they have the same, I don't know, ribose sugar molecule, the same phosphate, the same, what am I missing? The nucleotide base? Well, think about that. Translation of messenger RNA occurs at the ribosomes, but the translation of messenger RNA into protein only happens when the ribosome reads the start codon, which is AUG. And we'll talk about that a little bit more, but that starts protein translation. Anything upstream of AUG will not be translated into proteins. So AUG does actually two things. It tells the ribosome, read the messenger RNA starting here, and then it tells the ribosome or whatever, messenger RNA, we're gonna see how does that happen. It puts on, I'll word it that way because that tells just doesn't work. It puts on the amino acid, the first amino acid, and AUG codes for methionine, at least in eukaryotic cells. AUG encodes for methionine. Any question about that? So AUG does two things. It tells the ribosome start protein translation here, and it also encodes for which amino acid will be started there, and that is methionine. And then the ribosome will read the messenger RNA, and tRNA will put in amino acids. We'll talk about that in a little bit. And that will continue until the ribosome reaches one of three stop codons. So when the messenger RNA codes for UAA, UAG, or UGA, that is a stop codon, and the protein translation will stop. Now let me talk a little bit more about codons. A codon is a group of three nucleotides in messenger RNA. So each codon is three nucleotides, and those three nucleotides will code for one amino acid. Any questions about that? So you have three nucleotides which code for one amino acid. Oh, and let me state, you probably should know the start codon. There's only one, AUG, and you should also know it codes for the amino acid methionine. You do not need to know the three stop codons. There's three of them, so you don't need to memorize those. Any question about any of that? If not, let's talk about protein translation a little bit further. Let me shrink this so we can see it. Protein translation occurs with codons. And so we look at the genetic code. Each codon codes for one amino acid. But there are only 20 different amino acids, at least in humans and most cells. And yet there are over 60 codons. So what do you do with more than 20 if there's 60 possible codons? Well, there are many amino acids that have more than one codon code for that amino acid. Let me blow this up now so you can see the table a little better. Like for example, there are 2 codons that code for the amino acid phenylalanine. And then for the amino acid leucine, or is it, no, that's leucine, not lysine. Leucine, there are 6 codons that code for this amino acid. There are only 2 amino acids that only have 1 codon. And the first is, of course, the one we already mentioned, and that is AUG, codes for the amino acid methionine, and also tells the ribosome to start protein translation here. Okay. And I don't ever remember the other one. Is it tryptophan? Yeah, it's tryptophan. Is the other amino acid that only has 1 codon. All of the other amino acids have more than 1 codon. So there's more than 1 codon for most of the amino acids. And this is known as the degeneracy of the genetic code. Any question about that? All right. So this slide is showing you protein translation. And let's just go ahead and start the video. It'll go over it.
Translation is the synthesis of a protein from an mRNA template.
Just a minute, that's awfully quiet on my end. I don't know if you guys can hear that.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. A Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, any questions? So let's go to the next slide. So protein translation happens after transcription. And the ribosome assembles on the messenger RNA. And you don't need to really know, but there's two ribosomal units and they assemble on the messenger RNA. And the tRNA is important because it's actually what reads the codon. Like AUG codes for the amino acid methionine, and the tRNA brings that in. So it brings in the amino acid, and then the tRNA actually reads the codon. It'll have, at least the correct tRNA, will have an anticodon, which is the corollary to the nucleotides in the codon. So AUG will then bind to UAC, and this is hydrogen bonding. Any question about that? So in reality, it's not the ribosome that is reading the messenger RNA, it is the tRNA. What the ribosome does is it holds everything together and then moves down the messenger RNA, and it has two slots in it for tRNA to come in. In the first slot, the first tRNA will come in, and as I stated, it has an anticodon, UAC, which will hydrogen bond to the codon, in this case AUG. And it'll bring in the correct amino acid, which in this case is methionine. There's a second spot in the ribosome that can take in a second tRNA, and it will have the anticodon that binds to the codon, and it'll bring in the correct amino acid for this codon. Any questions about any of that? All right. The first amino acid is then removed from the tRNA and added to the second amino acid, which is still attached to the tRNA. This removal and then addition of that amino acid to the later tRNA amino acid. So this will be what's going to be the growing polypeptide chain right here. This removing and then addition happens by an enzyme, and that enzyme is actually an RNA portion of the ribosome. So this is the only RNA enzyme we will ever discuss in this class. You don't need to know that the first site of the ribosome is called the P site and the second an A site, because you can just call it the first site and the second site. Now, with the amino acid off of the first tRNA, it falls out of the ribosome. And then the ribosome moves down one codon. So what used to be the second codon, what am I saying, ribosomal position, will now move to the first ribosomal position. And then that'll allow another tRNA to come in to the second ribosomal position. Did that make sense? Once again, the amino acid on this tRNA will be removed, which actually is 2 amino acids now. This is the growing polypeptide chain. Will be removed and ligated to the newly added amino acid. And as I stated, that is the rRNA portion of the ribosome doing that enzymatic activity. This second tRNA will then fall out. Let's see if I got that on the next slide. Falls out, and then the ribosome moves down to the next codon, which isn't shown here, but it's shown there. So the growing polypeptide chain is always added to the last amino acid that was brought in. And then the ribosome continues moving down the messenger RNA with new tRNAs coming in, bringing the correct amino acid. And that is how the growing polypeptide chain grows. That process continues until the ribosome reaches a stop codon. Once the ribosome reaches the stop codon, the ribosome stops translating the proteins, no further amino acid is added. The growing polypeptide chain is then released. So that's the final amino acid. So that's now a protein. And then the ribosomal units fall off of the messenger RNA. And that means the messenger RNA is free again for a ribosome to bind and then move down the chain and start translating the protein, or the growing polypeptide chain would be a better way of wording it. Once the ribosome comes to AUG on the messenger RNA. Everything upstream of the AUG is not translated into protein. And as you know, the growing polypeptide chain, once it first is released, all you have is the primary structure, but the nearby amino acids will start interacting with each other. So we'll quickly get secondary structure, and then eventually we'll get tertiary structure. And if the protein is made up of more than one polypeptide, we will get to the quaternary structure. Any questions about any of that? All right. There is a complication of protein translation, especially in prokaryotes. There is a complication a little bit in eukaryotes, and it's showing you right here, DNA being transcribed to messenger RNA, and then the messenger RNA being made into protein. Anyways, what you're seeing is that this one messenger RNA can have more than one ribosome bound to it. And so you can get more than one protein being built at the same time from a messenger RNA. And it looks like we have 1, 2, 3, 4, 5, 6, 7, 8 different ribosomes on the messenger RNA making 8 different proteins. So that problem you can even have in eukaryotes. In prokaryotes, we have another problem, and that is protein translation is happening in the cytoplasm, but so is messenger RNA transcription happening in the cytoplasm. So you can have, let's see, how do I word that? You can have protein translation occurring on the messenger RNA before the messenger RNA is completely transcribed. And here you're seeing DNA, and there's a RNA polymerase here, here, and here, and here, making 4 different messenger RNAs. And this is the smallest because this is the gene, and the RNA polymerase is moving that way, so that's the longest. And then you can see that each of these messenger RNAs can have multiple ribosomes attaching. And it looks like on this one, there's 1, 2, 3, 4, 5 ribosomes making 5 different proteins on this one messenger RNA. And there's 4 ribosomes on this messenger RNA, which isn't finished yet, meaning the messenger RNA has got to be finished made. It's still shorter than it should be. And the ribosomes are already beginning protein translation. And even on this one, which is less than half of the size of the messenger RNA, we have 2 ribosomes on them, beginning to make proteins on a messenger RNA, which is still very small and has the majority of the messenger RNA to be made. Any questions about this complication? It's not a problem because the messenger RNA will get made with time. And then the ribosomes take time with the tRNAs to make the proteins. And so that's why in prokaryotes you can have both transcription and translation occur at the same time. Now, in a eukaryote, transcription happens in the nucleus and translation happens in the cytoplasm. So you would not have that happening at the same time. But you can have that same, we'll do it this way. You can have one messenger RNA have more than one ribosome attached to it, making more than one protein. Any questions about that complication? There's another complication in eukaryotes, and that is the DNA of a eukaryotic gene codes for regions that make the protein, but it also codes for regions of the DNA that do not encode the protein. An exon is a region of a gene that codes for the gene product, which in this case would be a protein. An intron does not code for the protein. And eukaryotic genes are that way. They have both introns and exons. Bacteria mostly have exons. And I'm not sure if there are any introns, but if there are, they're very rare. When the messenger RNA is first made in a eukaryotic cell, the exons and the introns will be transcribed. This initial RNA transcript cannot leave the nucleus. There is a gateway in the pores of the nuclear membrane, and the messenger RNA that has the introns cannot leave the nucleus. What happens is the cell cuts the introns out of the messenger RNA and then puts the messenger RNA pieces, the exon pieces, together. That cutting and rejoining is called splicing. So you splice out the introns and then splice together the exons. This messenger RNA that only includes exons can now leave the nucleus and go into the cytoplasm and be translated into protein. Any question about that? As I stated, this RNA processing only happens in eukaryotes. For all purposes, you can say that introns are only in eukaryotic genes, and the intron of the RNA will keep that RNA from leaving the nucleus. All right, if there's no questions, we're now going to move from talking about the structure and function of the genetic material to talking about regulation of genes. And we're only going to look at it at bacterial gene expression, but please understand that expression in eukaryotic cells is very similar. And how that gene expression occurs is by the operon, or operons if we're talking about several genes. And then after talking about gene expression, we'll move on to mutation, and then move on to genetic transfer and recombination, and then lastly close talking about genes and evolution. Any question about any of this? Well, bacterial cells like to regulate the contents in their cells. And one way to regulate a cell is to control the gene expression. For example, if you don't want a protein to be made, you can just turn off the gene, shut down gene expression. There are different genes in a cell. Constitutive genes are expressed at a fixed rate. So if you're making a constituent enzyme, that gene will always be turned on and will be made at a fixed rate. There's a name for those genes, housekeeping genes, but the name isn't terribly important. There are other genes that are expressed only when they are needed, such as an enzyme that is made only when it is needed. We will talk about the enzyme lactase, and this is the enzyme that digests the sugar lactose. Well, in E. coli, if the sugar lactose is not around, why would you make the gene product, meaning why would you make the lactase gene, the lactase enzyme? And cells decided a long time ago, I guess, that if they don't need a gene, they'll just shut it off. there are two ways to control gene expression. There's more than two ways. The two ways we're going to talk about. There's a repressible gene, and that gene is shut down when a signal is given. And there are inducible genes. They're normally shut off, but they can be turned on when the gene is needed. So there is regulation of genes, and we're going to talk about regulation by pre-transcriptional control, meaning you're regulating the gene product or the gene before transcription. And we'll talk about it with an inducible gene or an inducible operon. And I haven't talked about operons yet, but we'll get there. There are repressible operons where the gene is normally turned on, and when the cell doesn't want it, you turn it off. We're not going to talk about that, and you're not going to be tested on it. There's another way to control gene expression, and that's by epigenetic means. We're not going to talk about it, and you're not going to be tested on that. And you probably should say good thing, because epigenetic control is not well understood and it's complex. And I could say that it's not really understood at all, but that's not true. It's at least understood in part. But what's going on is not completely understood for epigenetic control. You can also control the contents of a cell by controlling the protein or by degrading the messenger RNA before the protein is made. And that is post-transcriptional control. We're not going to talk about that, and you're not going to be tested on it. Any questions about any of that? All right, so let's talk about an inducible operon. This is a gene which is normally turned off, but it can be turned on when the cell needs the gene, meaning needs the gene product or the protein. Any questions about any of that? And let me state that it does make sense for a cell to turn off and on a gene, because if the gene is not needed, why would you make proteins? They're not going to be used. All right, so I've already talked slightly about an operon. Let me clearly define it now. An operon does contain a region that we normally call the gene. And that has a specific name it's called the structural gene. Usually when we're talking about a gene, we are referring to a structural gene. Like when I was talking about the blue eye gene, I'm talking about the structural gene that encodes for blue eyes. Or when I'm talking about the gene coding for the enzyme lactase, that is a structural gene. An operon can be thought of as a super gene. An operon may contain one or more structural genes, at least in prokaryotic cells, and it contains the DNA nearby, which acts as controlling regions for the structural gene. Is that clear? So let's give an example. Z on the lac operon codes for the enzyme lactase. So this is the lactase gene. Now, in the lac operon, there are 3 structural genes on the operon. The genes that are on the same operon are always related to each other. In the case of the lac operon, Z, as I mentioned, codes for the lactase enzyme. Y, the gene Y, is a structural gene that codes for the transport protein that brings the sugar lactose in the cell across the cell membrane. So the Y gene codes for, it's called permease, you don't need to know that. Sorry, I'm drawing a blank here. The group, not the group, I almost had it. Transport protein. So Z codes for the enzyme, and then Y codes for the transport protein that brings the sugar lactose into the cell membrane. And then A codes for, or I should say, helps code for converting the sugar beta galactose into the sugar glucose. There, I said it right. So all 3 genes are involved in the metabolism of the sugar lactose. The Z gene degrades the sugar lactose into fructose and sucrose. The Y gene brings the sugar lactose into the cell. And then once the sugar lactose, oops, I didn't say that right. The lactase gene breaks the sugar lactose down into glucose and beta galactose. And then the A gene helps change the sugar beta galactose into the sugar glucose. So all 3 genes are involved in the metabolism of lactose. Since we're talking about structural genes, this is the structural gene of a bacteria. You should know that the structural genes of an operon of a eukaryotic cell contain only 1 structural gene. Now, those of you who drink milk know that you metabolize the sugar lactose. So you got the Z gene on the lac operon, and you do have the Y gene to bring in the sugar lactose into your cell, and the A gene for converting galactose to glucose. It's just that you have 3 operons, 1 structural gene for each operon. And that is how eukaryotic operons differ from bacterial operons. There is one other difference, and that is eukaryotic cells have a lot more control elements for controlling the expression of the gene on the operon, meaning the structural gene in the operon. Any questions about that part? If not, the operon also contains controlling regions of DNA that regulate the expression of the structural genes in the operon. There's the operator, which we'll talk about shortly, and the promoter, which we've already talked about. And then there's also the terminator after the last structural gene. Any question about any of that? In the case of the lac operon, there is another gene, which is a regulatory gene, and it controls the expression of the lac operon, meaning it controls the expression of all the structural genes in the lac operon. This regulatory gene is not part of the lac operon. It is its own operon, meaning it has its own structural genes, or gene, probably just gene in this case. And it also has its own controlling regions as part of its operon. The regulatory gene controls the expression of the gene of interest, in this case, the lac operon. What else did I want to say about that? Normally the regulatory gene is not near the gene that it regulates. For example, on eukaryotic chromosomes, the chromosome that has the operon on it usually does not have the regulatory gene on it. It's usually on another chromosome. Any question about any of that? All right, so how does the lac operon get turned off? Well, the regulatory gene, which happens to be close to the operator or operon it works on, makes a repressor protein. This repressor protein is active, and it will bind to the operator of the lac operon. Now, when the RNA polymerase binds to the promoter and starts moving down the DNA, it'll bump into the repressor protein, and the RNA polymerase cannot move further. It's stopped by the repressor protein. That effectively shuts down the lac operon because the lactase gene will not be transcribed. Any question about any of that? Normally, in the case of E. coli, the repressor protein is made, it binds to the operator, and then it shuts down the lac operon. Why in E. coli would E. coli normally shut down the lactase enzyme? Why would it shut down the lac operon? Anybody gonna guess? Well, let me ask an easier question. How often does E. coli find the sugar lactose? What do you know about the sugar lactose? Well, the sugar lactose is not normally around E. coli. There is an exception, but we'll talk about that whenever you guys are willing to talk. So E. coli normally shuts down the lac operon because E. coli is not normally going to see the sugar lactose. Well, what happens when the E. coli needs the lactase enzyme, meaning the E. coli is going to see the sugar lactose? The gene gets turned on. How that happens is that the repressor protein gets made like normal, but then it binds to the inducer. The inducer is allolactose. Allolactose is an isomer of the sugar lactose. So whenever you have the sugar lactose around, it gets converted to its L form. If you don't remember, an isomer is the same molecule. It's just one is left-handed and one is right-handed. And so whenever lactose is around, its isomer is around. And then allolactose will bind to the repressor protein. When it binds to the repressor protein, it inactivates the repressor protein. Once it's inactive, that repressor protein cannot bind to the operator. And now when the RNA polymerase binds to the promoter, it moves down the gene and transcribes the lac operon. Okay. Well, here's another question for you. What about this protein that was already bound to the operator? How does it leave the operator so that the gene can get turned on? You guys really should participate a little. What happens is that the operator and the repressor protein, this binding is not permanent. It's a stoichiomic reaction, and every once in a while, the repressor protein will fall off. Now, if there's other repressor protein that's active around, it will bind to the operator, or the same repressor protein will bind to the operator, and that will keep the lac operon turned off. But if the inducer, the allolactose, is around, it'll bind to the repressor protein and inactivate it. And now all of the repressor protein is inactivated. And so that is how the lac operon will turn on. The repressor protein just every once in a while falls off. The same happens with this polymerase right here. When it binds to the promoter and then moves down the DNA and bumps into the repressor protein at the operator, this RNA polymerase doesn't get stuck here forever. It binds with a stoichiomic reaction. And so every once in a while, it'll fall off. And then the RNA polymerase can go on and transcribe another gene. So it's not always tied up. Okay. Anyways, that is how the lac operon is turned off by the repressor protein, and how it's turned on by the inducer, which is an isomer of allolactose binding to and inactivating the repressor protein. That leads to the transcription of the lac operon. And what do you notice that's unusual about this messenger RNA? Come on, I know you guys know this one. All right, think about that. Anyways, the lac operon is only turned on when it is needed. Once all of the sugar lactose is metabolized, the repressor protein will become free because the binding of allolactose to the repressor protein isn't permanent. So every once in a while it falls off too. And now there's no more lactose around, so there's no more allolactose around. And so that repressor protein can now bind to the operator. And it makes sense for a cell to do this because the cell would not want to make the enzyme lactase if the sugar lactose were not around. But it would want to make the sugar, would want to make the enzyme lactase if the sugar lactose was around. Any question about any of that? All right, so that's it for gene expression. You should realize that we did discuss that in a very simple way, meaning I told you the gene was off or the gene was on. In reality, there's different levels of being on. You could have a gene that's barely transcribed at all, one that's slightly transcribed, one that's moderately transcribed, and then one that's heavily transcribed. And there are mechanisms to control that too, but we're not going to get into it. Suffice it to say, when the E. coli or other cell needs a sugar, and if allolactose or lactose is around, then the lac operon will be turned on. And if it really needs a sugar, meaning glucose is low, it'll turn on the expression of the lac operon higher because the sugar is needed. We need to metabolize it. Any questions about any of that? If not, let's go ahead and talk about mutation. A mutation is a change in the genetic material. You simply need to change the DNA and you have the mutation. Mutations may be neutral, meaning they have no benefit or not. They may be beneficial, that the person who has that mutation gets some benefit out of it, or the mutation could be harmful. Most mutations are either neutral or harmful. A mutagen is an agent that causes a mutation. I should say increases the mutations. Spontaneous mutations can occur, and they occur in the absence of a mutagen. Simply by the DNA replicating itself every once in a while, not very often, about 1 in 10 to the 9 times, the wrong nucleotide will be added. And if that's not corrected, it will lead to a mutation. So spontaneous mutation can occur, but it happens at a low rate. And here we're looking at a mutation in the person's eye. There are different types of mutations. Here we're looking at a possible mutation where metal spoons would stick to the boy's skin. And last I heard, they were looking at this child to try and determine why that was occurring. They were wondering if it was, he had some magnetic field and was attracting metal spoons to his skin. So he was like a baby Magneto, if you know what that character is. Or was it simply that his skin was really sticky and so the spoons would stick to his skin? And I'm afraid I only read the article, but they were looking into it. I was never able to find an update where they were talking about what they found. But there are different types of mutations. There are a base substitution, and we can call that a point mutation, where one nucleotide in the DNA is switched or changed. There's 3 types of base substitutions. There's a silent mutation, a missense mutation, and a nonsense mutation. We'll talk more about those in a little bit. There's also a frame shift mutation. This is not where the base is changed. It's where an extra base is either added or removed in the DNA. So you get an extra nucleotide added to your DNA. That's a frame shift mutation. We'll talk a little bit more about that also. I should mention that the addition could be more than one nucleotide. It could be 2 nucleotides, 3 nucleotides, or more nucleotides. They're all called frame shift mutations. Although if you add 3 new bases or subtract 3 new bases or some multiple of 3, you don't cause a frame shift. And I'll explain that later. And so those are not a frame shift mutation if you remove by a multiple of 3. Why it's not a mutation if you pull out 3 nucleotides? It's because you removed that codon. But the next codon will code for the protein. So the protein is changed, but it's lacking one amino acid. It doesn't have new amino acids. And I'm just going to explain it that way for now. We'll get to that in a little bit. There's also another type of mutation which is not discussed in your book. So you need to pay attention closely to this lesson and to my notes. For some reason, the textbook author did not talk about the third type of mutation. It is a macromutation, where the entire region of a chromosome is either added or deleted. And you actually probably know a macromutation that has the entire chromosome added. Does anybody know what that is? I'll give you a hint. These individuals have an extra chromosome 21, and that is a macromutation. Nobody's going to guess? I think you guys figured it out. You're just not talking. All right. I think this is a good place to stop. Oh, wait a minute. We don't have a lab today, so I can continue going on. And that's probably good because we are a little bit behind schedule. Slide 43, if I lose it. Yep, I lost it. So let's talk for 10 more minutes. A base substitution occurs in DNA replication where the wrong nucleotide gets added. So here we're using this strand as a template to make the new strand, and the RNA polymerase added the wrong base here. It should have added a C, and instead it added a T. If that is not corrected, when this strand gets replicated, it will change the nucleotide pair. And that will cause, in this case, another protein, another amino acid for this protein. So we will mutate the protein. So normally we get the codon being UGU, and it'll be switched to UAU. Once this happens, the cell could find that and correct it, meaning remove the T and then add a C. So this mistake does not always get made into a mutation. But if that T is ever replicated, you get the mutation, and then there's no way of reversing that because the wrong nucleotide is on both strands. So once the DNA is replicated, this mistake becomes permanent. Any question about any of that? All right. Let's talk a little bit about mutations. We mentioned that a mutation can be beneficial, meaning it improves an individual better. And I only know of one example off the top of my head, and it was for fruit flies where they were in a bottle. I'll pretend this is a bottle. Let's see, can you even see this? No, because it's the wrong color. Let's see if you can see this side. Not until I put it by my face. You can, the beneficial mutation was the fruit flies put their pupa on the sides of the glass container. And then when the biologists were growing them, they noticed that there was a mutation in the bottle of fruit flies, and that mutant allowed the fly to put the pupa on the cotton swab, which was the top of the container, as well as you could put it on the glass sides. So that allowed that mutation to put the pupa in more than one location. So that was beneficial for that fly, because when all of the space became occupied, meaning all of the glass sides became occupied in the container, the other flies could not form a pupa. But the one that was a mutant could put it on the top of the container and could continue putting it on the top of the container. So that individual increased in numbers in the population. And I think, I don't know if they followed it, but that individual eventually took over the population. So that all of them, or almost all of them, did that. So that's the only beneficial mutation I can think of. You know many examples of a harmful mutation where a child is born with a mutant gene and it's defective. And so the child has a disease like Tay-Sachs disease or hemophilia. Those are harmful mutations. Okay. There's also a silent mutation, and you can call that a neutral mutation. And that's where you change one nucleotide of DNA, but that changes the codon to another codon that codes for the same amino acid. For example, if UUU was mutated to UUC, that still codes for the amino acid phenylalanine. So the protein is not changed. And that is called a silent mutation. It is neutral in a population, meaning someone who gets that mutation, it's not beneficial, it's not harmful, it doesn't change the protein at all, and so it's neutral, no effect on protein. A missense mutation is where you change one nucleotide base and it changes the protein, it changes the amino acid that the codon will code for. For example, nope, it's not that one, it's actually this one. The DNA is CCG and it gets mutated to TCG. That changes the amino acid from glycine to serine. And so the protein is changed. This protein may not fold correctly. And so it may have less activity. Anyways, the point is a missense mutation changes one base, which results in a change in an amino acid in the protein. Some examples of this missense mutation is sickle cell anemia, where we change one protein in hemophilia, which is a protein with thousands, did I say that wrong? You change one amino acid in a protein that has thousands, maybe hundreds of thousands of amino acids. You only change one, but it changes the protein fairly drastically, and then the individual will be sickle cell anemic if they have two genes for sickle cell anemia. And that will be selected against, at least for the individual that has two sickle cell anemic genes. Another example, which actually happened in the human population about 10,000 years ago, is we had a simple single nucleotide base change resulting in a missense mutation for human eye color. Before that time, every individual in the human population only had brown eyes. And 10,000 years ago, there was a mutation that resulted in blue eyes. And now the human population has both blue eyes and brown eyes. And there are different variations in color, okay? But anything that's bluish has the blue eye gene, and anything that's brownish has the brown eye gene. So there are other genes that can alter the color somewhat, but they never change it from blue to brown or brown to blue. Anyways, that happened about 10,000 years ago. And that gene increased in frequency, at least in Northern Europe. So it's now the predominant gene in Northern Europe. When we look today, and obviously we don't know in the past, but when we look today, there is no benefit for having the blue eye gene. In the past, there either was a benefit or the blue eye gene was next to a favorable mutation, and the favorable mutation increased the blue eye gene in the population. Any question about any of that? All right, we're 20 minutes out, so I'm going to stop right here and ask, are there any questions? If there's no questions, I will see you next Tuesday. Bye.